Hello and welcome to Unit 1.2 Carbohydrates. This is a short revision video. If you would like a longer, more detailed version of this video, then check the top link in the description. You can also find other resources such as flashcards, detailed notes and revision websites in the description. I will now go over the key points of Unit 1.2 Carbohydrates. To recap, monosaccharides are the monomers from which larger carbohydrates are made. Glucose, galactose and fructose are common monosaccharides. Disaccharides are formed by the condensation of two monosaccharides. Glycosidic bonds are formed. Maltose is a disaccharide formed by condensation of two glucose molecules. Sucrose is a disaccharide formed by condensation of a glucose molecule and a fructose molecule. Lactose is a disaccharide formed by condensation of a glucose molecule and a galactose molecule. All monosaccharides are reducing. All disaccharides except for sucrose are reducing. Glycogen and starch are formed by the condensation of alpha-glucose. Cellulose is formed by the condensation of beta-glucose. Glycogen and starch are both storage molecules with 1 to 4 glycosidic bonds between molecules of alpha-glucose. They also have branches which make it easier for hydrolysis as there are more ends for enzymes to attach. This allows them to be broken down quickly. There are 1 to 6 glycosidic bonds at the branches. Glycogen is the animal storage molecule and so has more branches for faster hydrolysis. Cellulose is a long, straight chain of beta-glucose and is used in cell walls. The cellulose chains are linked together by hydrogen bonds to form strong fibers called cellulose microfibrils. There are only 1 to 4 glycosidic bonds. The Benedict's test can be used to check for reducing sugars. A color change from blue through to red means reducing sugars are present. To test for non-reducing sugars, take a fresh sample of the solution and either heat it with diluted hydrochloric acid or hydrolyze using enzymes. You can then neutralize it with sodium hydrogen carbonate. Once you have done these, you can continue the Benedict's test in the same way you would for a reducing sugar. To test for starch, just add iodine dissolved in potassium iodide solution to the test sample. If there is starch present, the sample changes from brown-orange to a dark blue-black color. In your exams, make sure you always talk about iodine dissolved in potassium iodide and not just iodine alone. Thank you for watching Phreology Revision.